membrane protein structure biology. So I'll quickly read her profile before she comes on board. So Shubham Dubey is a graduate research assistant at Noinaj Lab, Department of Biological Sciences, Purdue University. She attained her BS in Botany, Institute of Science, Banaras Hindu University, India, and her MS in Biotechnology, Indian Institute of Technology, Rocky, India. She's also doing her PhD in structural biology and computational physics at Purdue University. She has attained several academic and professional honors, some of which are American Heart Association Pre-Doctoral Fellowship, Women in Science Program Travel Award, Purdue College of Science Travel Award. And she's going to be talking to us this evening right now. Right, so can you all join me as we welcome Shubham to take our presentation?
Can you hear me?
Yes, we can.
Okay, awesome. Thank you for the kind introduction. I am trying to share my screen now, and if you can just quickly confirm if you can see my screen, that would be great. Okay, can you
All
all
right. Yes.
see my screen?
Yes, we can.
Okay. Okay. Um, Thank you. Thank you for the kind introduction. Hello, everyone. I'll be giving a brief introduction. I'm not talking specifically what I'm doing in my research here at Purdue, but I'm going to give you an overview how single particle cryo-EM has revolutionized the membrane protein structure biology. So before we dive into the topic, I want to give you a brief introduction of the membrane proteins itself, because many of you might not be working in the field, so it would be hard if I describe the problems and the solutions that cryo has come up with for the membrane proteins. What do they mean in terms of how membrane proteins generally look like? So membrane proteins are just like the protein molecules, but they are situated in the membrane, and depending on their locality or how they are tethered to the membrane, they can be categorized in different classes. They can be integral membrane protein, which means that protein is totally embedded inside the membrane. In that category, there can be two types, transmembrane protein or membrane associated, depending on if protein travels to both the bilayers. You can all see my marker pointer. Um, depending on if they travel all the way, both leaflets inside the membrane, they can be transmembrane protein, or if just they are tethered to the one part of the leaflet, they can be membrane-associated protein. The another class of the membrane proteins are lipoproteins. These are the proteins which are not directly linked to the membrane, but they contain a lipid anchor that tethers the protein to the membrane surface. And then you have this another kind of membrane proteins called peripheral membrane protein. They are not directly again associated with the membrane, but they attach with another protein which is situated in the membrane, and that's how the protein-protein interaction holds these proteins to the surface of the membrane. So this is the kind of like a brief introduction of types of membrane proteins that exist in biological system. But there is one class where the membrane itself changes, which is the interest of our lab, which is the outer bacterial outer membrane proteins. So these are the beta barrels, and also if you compare the membranes like side by side, you see that the inner leaflet is pretty much the same in both the membranes, but the outer leaflet is different in case of bacterial outer membrane protein. So you see that there is a lot of diversity within the protein and also the membrane. They both are associated with the membrane protein. So I hope I gave you a brief introduction of the membrane protein by now. So before you even think about characterizing your membrane protein, you need to understand from where the membrane protein is coming from, because I told you that the membrane matters, membrane can be different. And depending on the origin of protein, you want to choose your expression system. If you are working on a bacterial membrane protein, you can choose E. coli, which is a very well known and well common expression system, which is used in multiple labs all across the world. If you're working on some mammalian protein, then you want to express them into the mammalian cell, because the membrane composition of the bacterial system and the membrane composition of mammalian system will be different, and it will be very high expecting of you if you expect a bacteria to express the mammalian protein in the bacterial membrane. So those are the things that you need to take care of mind. Other than that, there are other expression systems such as yeast, insect cells, or if you're working on plant cell, you can also use feed germ cells. uh, so depending on how protein is where protein is originating from, you want to choose your expression system based on that. So the very first step of getting a protein can be very challenging for membrane protein if you're working in that field. So once you get a good expression system where your protein is actually expressing, then comes the purification part. So there are mainly two methods to purify the membrane protein, but this is a very bird eye view of the methods. Inside the method, there is a lot more complexity associated. So this is the general pipeline. If anyone is working with the protein expression system, they know that you express the cells and then you lyse them and then you centrifuge them and then you take the supernatant out. Once you take the supernatant out, that supernatant has membrane fraction because you have lysed the membranes over here, but you also have the soluble protein fraction in that supernatant. So then the next step, which is um, exclusive to the membrane protein purification, comes the ultracentrifugation. You do the ultracentrifugation there, that pallet downs the membrane in the bottom of the tube, and then that fraction contains only the proteins that are situated in the membrane. But now you have to take the membrane part separate and the protein part separate, and that's when the detergent comes into the play. And then you solubilize the membrane fractions into the detergent, and then you follow the rest of the purification protocol. So each of this step that I'm talking about will be very modifiable depending on what kind of sample you are working with. And detergent is one of the most crucial one, because you want to scan a lot of detergent that works for your protein and that can solubilize the membrane protein that you are working on. And it's not degrading the protein. Meanwhile, you are extracting them from the membrane. So a lot of optimization goes on at this particular step, which I'll be coming on in a bit. And then just let's assume you are unlucky enough and then your protein never goes to the membrane. If it never goes to the membrane, cell has a rescue system that it sends the protein into the inclusion bodies. Inclusion bodies contain the protein, but they are not folded and they are not present in the most native environment because it needs membrane around it. So you unfold the protein in 8 molar urea, and then you try to refold it over the night, doing a dialysis, and then you follow the further purification step. I'm showing you here everything in a ray diagram, but it's not that easy because each step will need further optimization. Like you want to do dialysis, then you want to do at what pH, what temperature, what urea concentration. So a lot of things goes on in the background, and then if that works out, then you get your pure sample. So once you get your pure sample, there were two methods that were available before single particle reconstruction, were cryo-EM and crystallography. We know that the crystallography gives really high detailed information about the protein molecule to subatomic resolution, and then you can do the biological research followed by getting the structure. And then you have another method, which is the NMR, solution-based structural determination method, where you can understand the dynamics of protein. But the problem is that both these methods, they need very high concentration of protein and they need highly pure sample. So given in the previous slide, I explained you how difficult it is to get the sample. Both these techniques are very demanding in terms of high concentration and getting a pure sample. So you really get a little bit of sample, and you don't get to try a lot of options that you can try with NMR and crystallography. And on top of that, getting a crystal of a membrane protein is another challenge. So um, so people started working more on the microscopy side. They tried improving the microscopy technique. So microscopy existed for a while where you can just take a lot of 2D images and then reconstruct a 3D image based on all those 2D projections shown here in this diagram. But if we compare before 2013, we were getting these blobby kind of structure in terms of resolution with the single particle reconstruction. But after 2013, we can really see the subatomic resolution of the molecule. And the credit goes to a lot of development that goes into the background of the technique itself. There has been a lot of improvement with the imaging hardware, such as improved gun with the microscope, good detectors, and then freezing techniques. How we freeze our sample that has improved over time, so we can freeze our sample in the vitrified before it can convert into the cubic ice. And then also a big credit goes to a lot of development in the software part, especially the CryoSpark, which has speeded up the um, processing of the data over time a lot, basically. So this all has worked with the soluble protein in the early development of the single particle reconstruction, but finally the it impacted the membrane proteins as well. And the first structure of the membrane protein was solved by the cryo-EM. Not going into the detail of the protein itself, but you can see that there is a blobby density where the helices are barely fitting in, and you know where things are situated with respect to each other, but you don't see much atomic resolution at even at this point. And also, one more thing that I am not that is not visible in this particular view. If you see from the top, if one corner of the protein, if you see one part of the protein, there is a symmetry that exists between the protein. So if you just see this one part clearly, you can apply the symmetry operator and you can reach to the good resolution. So earlier structure, basically, they relied on this symmetry operation to get to the high resolution. But in biological system, this is very rare that a membrane protein will all the time will have some kind of symmetry to reach to the high resolution. So what people come up with is if you collect the large amount of data set in diverse orientation, these are just the 2D classes that I'm showing from my own research, you can see that each particle is situated in the different orientation. And even getting in different orientation, when I collected the smaller data set, I was able to reach to the resolution, which was roughly like 4.5 angstrom resolution. And you can see that the helices are little bit clear that where they are situated. But if you see the beta sheet, they look like a sheeted barrel. There are no discrete beta sheets that can be visualized at that subatomic resolution. So then I collected on the same sample, I collected more data sets, and then I was able to reach to the resolution of 3 angstrom, and where you can see clearly now the beta sheets are visible. So yes, you can see here an example how collecting more data, like first the single particle reconstruction has helped getting a membrane protein structure of such large biological assembly, and also helped us reach to the 3-ish angstrom resolution with collecting more data and having different orientation of the particles on the grid. So previously, when I was talking about the expression challenges and purification challenges of membrane protein, I described the detergent is very important. So what people do is they basically go into the literature and they try different detergent that has been tried before for the given class of membrane protein, and you just have to like scan it through, and then you have to go what works best for your sample. Even if you work out that the protein is solubilized in that particular detergent, it is stable in that detergent, it might hinder with your microscopy step, because they reduce the contrast of the images. Because if I show you here, these are the big micelle structure. So these round structures are basically the micelles around my protein. So if they are giving a lot of background into the images, then I won't be able to see the features in that particular area. So they reduce the contrast. That's one thing. They can form a big micellar structure, which can be bigger than protein itself. So you don't know where the protein is situated inside the micelle. you will have really hard time aligning the particles one on top of each other and forming a 3D map out of those particles. And then sometimes even they induce artifacts into your sample. They represent a state of the protein which is actually not true, and it doesn't exist in the biological system. So to overcome these problems, we have some solutions in the field currently. You can use amphifolds. Those are polymers with the empiphatic empiphatic uh, characteristics. And you can also use nanodisk. I'll come to the nanodisk in a bit. And then SMA particles, and also you can use liposomes. They not only solubilize your protein in the detergent-free environment, but they give a more native environment to your protein. So you get a membrane protein orientation or oligomerization state, which is more natural in the biological system, not the detergent-induced artifact. And here is an example to show you that the artifacts induced by the detergent are real in membrane protein samples. Is that when this protein was reconstituted into the detergent, you can see from the 2D class itself that the protein is a dimer. So if a person looks at this image at the first, it will make a claim that the protein is a dimer, and it functions as a dimer. But what happened that the same protein was reconstituted into the nanodisk, and this time it was a monomer. So and later on it was also solved in liposome and a lot of other kind of detergent substitutes, and it was a monomer basically. So here you can see that detergent clearly wasn't using an artifact into the sample, which was cleared by the nanodisk later on. And I kind of convinced you that the nanodisk is the solution for everything. It's actually not true. It just depends on your sample. And here I'm showing you an example of a membrane protein solved in different detergents, such as GDN, LMNG, digitonin, and also solved in two kind of nanodisk made up of MSP2 and MSP1. And you can see the resolution is going bad in this kind of MSP. And also this detergent is not giving a good resolution. While GDN, which is a very large micelle detergent, it's giving good resolution for that protein. So basically you try everything, and whatever works is the solution. So you have to really try all the possibilities and scan all your available resources. And another interesting hurdle in the membrane protein single particle reconstruction, or you can say in general for single particle reconstruction, is the size. You will be hearing a lot about this in the workshop throughout, but I'll just give you a brief overview how we overcome with the size problem in membrane protein biology field. Is that either you combine it with the other partners of the protein which it interacts with, so that the protein becomes a little bit bigger, or you add antibody fragments to your membrane protein. Like this, in this membrane protein, the membrane part is this much, but you are adding the extra molecular weight with this fab-like molecule, so that your overall size of the protein becomes bigger, and then you can solve the structure of the protein. Because if you just try to solve with this part will be totally covered with the micelle or your nanodisk, and you will not be able to align the particle pretty much. So this acts as a fiduciary marker in your sample, and you will be able to solve even the structure of even the smaller membrane proteins. So um, it's still the size is a problem, but we are getting there. So I think it will advance more and more and in upcoming years. And with that, I think I'll be happy to take any question. I hope I gave you an overview how membrane protein got advanced over time using this method. Thank you very much.
All right, thank you so much, Shubham, for that presentation. She really made us see the application of Cryo-EM in membrane protein, right? Yes. So we'll be moving on. Questions. Can we have questions, please? Can we have any questions in the chat box? All right. Angana is saying wonderful presentation. All right. It seems there are no questions. I guess people are
I think
tired.
I received one in my
I have a
chat.
question. I have a question.
All right.
Yeah, go for it.
Question.
Mm-hmm.
Oh, where do I go to? I imagine.
All right, you can proceed, ma'am.
Okay, so thank you very much for your presentation. I want to find out membrane proteins, when in trying to isolate and purify a particular membrane, we normally have contamination from other membranes because we have the plasma membrane, we have the internal membrane, that's membranes of other organelles in a particular cell. Now, how do we isolate the particular membrane we're interested in? For instance,
So,
if I want to study the membrane of mitochondria, I will have contamination from membranes of other organelles within the cell.
yes,
How do I isolate just mitochondrial membrane to study?
That's a very great question. I think the best approach, working with any kind of, so cell has different organelles, right, inside it, and every organelle, like mitochondria, chloroplast, they all have their own membranes, right? So what you do in the first step is look at the centrifugation speed on which you can just centrifuge that organelle first. So first lyse the cell and then centrifuge that organelle, and then after that organelle, even in mitochondria, you'll see that outer membrane, there is an outer membrane and there is an inner membrane. So then you have to decide, so basically where is your protein situated? If it is in outer membrane, then you add a detergent called Triton X. You can look into the literature. There are other detergent as well that if you put those in detergent for certain amount of time, they solubilize your outer membrane. Once you solubilize your outer membrane, then you can do in the next step another centrifugation. Then you can get rid of the all inner membrane part and the cell debris inside the mitochondria one. And then the given membrane will contain just the outer membrane of your mitochondria. So basically, that's why I was saying that till the point of lysis, it's fine. But when it comes to the ultracentrifugation, you have to really look where your protein is located and how much speed and how many times you want to do the ultracentrifugation by using that particular detergent is the step for the optimization. So I think the quick advice I can give you is that first lyse the cell, then centrifuge the mitochondria first, check the speed on which the mitochondria centrifuges, and then lyse the mitochondria outer membrane, and then do another ultracentrifugation where you can separate your mitochondria outer membrane and inner membrane depending on where your protein goes. If it is an outer membrane one, you centrifuge the outer membrane. If it is inner membrane, then you know which fraction to look for. I hope that
Okay. So it's
answers your question.
fractionation based on their densities.
Yeah. Mitochondria goes
It's
in a
heavier.
way
So
different density. Yes.
yeah, and because it has more proteins because of
Mm-hmm.
enzymatic activities.
Right. So yes, when you're lysing, make sure you add good amount of proteases because mitochondria goes a lot of oxidative damage and a lot of other things. So um, keep it in the most protease inhibited environment, and also um, try density centrifugation because you know which layer your mitochondria sits in.
Yeah, yes.
Then take that layer and then do
And
the purification further. If you're doing an assay and stuff, then maybe the amount of protein won't be enough. Given I don't know the purpose of the protein, but you have to really start with a very big batch of cells, and then you will end up with some mix of protein by the end. Basically, you will lose the protein at each step. So you have to really start with a big batch because when I purify my protein, I start with the 24 liters of culture, basically. And I end up with few mix of protein where I can just set some grids with it. So you have to work out the odds in that scenario.
Okay, thank you very much. Secondly, for
Mm-hmm.
integral proteins, yeah, you made mention of the use of detergents. In fact, today I've learned that liposomes can be used as detergents for isolation. But what about peripheral proteins?
So
Peripheral
no,
proteins,
no,
how do
no,
you?
don't get me wrong. Don't get me wrong there. Sorry, if I badly explained it. So first you take the membranes out, then you solubilize it in your detergent, and then you reconstitute that into your liposome. The liposome don't come as a substitute when you are
Okay,
taking the membrane protein out from the membrane. First, you have to go with the detergent. You have to find the best detergent, and then you have to replace your detergent with the liposomes.
lipoproteins, okay. What about peripheral proteins?
Same
How
way,
do
same
we
way.
isolate peripheral
And
proteins?
some of them, like even in the peripheral protein, what you can do is if you don't think that particular domain is very important uh, for the science that you are looking forward to, you can just delete that particular domain and treat the rest of the protein as a soluble protein and work with that.
Okay,
Yeah,
thank you very much.
you're welcome. Thank you. Thank you for the question. I have a lot of questions in my DM. The first question is by Chandan Vishwakarma. He is asking that does cryo help to study change the orientation of the protein? Does cryo help to study the change in the orientation of the protein? Uh, uh, I if I got your question right, Chandan, what means change in the orientation? Yes, because um, like in one of my own personal cases, protein N lobe interacts with the C lobe, and in the other state, the protein C lobe and C lobe interaction happens with the other partner. So yes, you can study the orientation differences, and nowadays with the advanced tools, you can actually see those transitions happening between your particles if you have a large number of data set where every step while changing the orientation is present on your grid. So yes, you can study the change of orientation. Hope that answers your question. Please, can you get your email address and share how research on the PhD student? Okay, sure. You can get my email address from Yomi, and I can send it to um I think I covered most of the questions. So over to you, Abby.
All right, thank you so much, Shubham, for that. I'm really glad that people were able to understand and follow and ask questions. We have, before you go, we have Professor Miriam Solomon in the house. She is the Dean of the Faculty of Basic Medical Sciences in University of Jos. We really appreciate your presence, ma'am. She was around earlier on in the at the venue of the program, but she had to leave to attend to some duties. So we really appreciate you, ma'am, for being around for this workshop. And thank you, Shubham, again for the presentation. We're really happy that you could do this out of your busy schedule and you could make this presentation for us. Thank you so much.
Happy to share the war that we go through every day.
All right.
Yeah, thank you. Thank you so much for having me.
Thank you so much. I also want to appreciate Professor Wang. He has been in this meeting from the beginning up till the end, up till now. He has been in the meeting, watching. He gave his lecture, and then he's been watching everything that's been going on. And like I mentioned earlier during the day, He's the director for the Cryo EM over at Purdue University. So he was the one who spearheaded the collaboration between Purdue University for this workshop to be holding. And we really appreciate you. Thank you to all our participants. Thank you for staying up till this time. We will continue tomorrow. I'm going to hand over to Professor Taiwo Alemka very soon. However, we'll continue tomorrow. Tomorrow, we are starting by 10 a.m. Nigerian time. Please kindly spread the word. It's 10 a.m. Nigerian time. We're going to have 3 sessions that will be both on-site and online, and then we'll have 1 fully virtual session. So for the first session, we'll be taking practical session as well as the lecture session. So we're talking about cryo approach for learning cryo EM technique, both hands-on, which will be hands-on for on-site participants, and then we'll do a lecture and another practical cryo EM data processing. For the third session, it will be cryo EM map analysis and model predictions. And for the last session, which will be the final session for the whole workshop, we'll be doing introduction to relevant databases for virtual screening, and then structure-based virtual screening, lecture and practical. So that's the one we know popularly as molecular docking. So we'll be connecting everything. It promises to be an awesome time tomorrow. You cannot miss it. This provides us an opportunity to see where the world is going to and have an experience with it. So I'll hand over right now to Professor Taiwo Alemka for the closing remark. Thank you.
Thank you very much, Ms. Adegboyega. It's a pleasure to be here. Wow, it's been a long day. And, one unique thing I would like to say about the opportunity of this workshop is that it brings us to a point where we are abreast with an emerging technique. We are used to playing catch up, especially as the saying goes, people from the global south playing catch up with the rest of the world. But now, this opportunity through this workshop is like no other. And honestly, I think, this is something quite unique. We may struggle a bit, but I think everyone is ready to learn, and it will be a worthwhile experience. And we're beginning to see that. I want to thank all the facilitators. They have tried so much to bring Cryo EM and enable us enjoy the virtual reality, in a way that it's well understood and we can also follow quite readily. It's a very unique privilege, and we want to thank you very much. We want to thank Jaris for making this happen. We want to thank all the participants who have stayed in all along from the beginning till now. And tomorrow is a day to look forward to once again. Thank you very much. It's also a pleasure to bring this closing remark. Thank you, and we'll see everyone tomorrow. Thank you very much.
Thank you very much, Prof, for the wonderful closing remark. Appreciate you for being there. That's Professor Taiwo Alemka. It's also one of my mentors. Thank you very much, sir. We really appreciate your presence. Okay, so we'll bring today's meeting to a close. right now as we converge tomorrow again for next session. However, I notice my prof is still in the house also, Professor Wen Jiang. If he's there, if he has any comment to make before we close, we would like to also take that. Okay, all right, so I guess, okay, please go ahead, sir. Yeah, go ahead. You are muted. Let me, okay, good.
Okay, yeah, okay, good. Yes, yeah. Okay, it's great. It's good success that we have so many participants on site and through Zoom that to listen to our introduction to Cryo and Cryo VR. I'm glad that so you have liked what we have done in terms of disseminating this Cryo as a powerful new method to you. Hopefully, you will get this as a good starting point, and you're welcome to contact Yomi and myself later if you want to get more information about this. We'll be happy to help. Enjoy your evening and enjoy next day's program. We'll see you later. Thank you.
Thank you. Thank you very much. Thanks, everyone. See you tomorrow. Yeah, bye for now.